At the University of California at Berkeley, psychologist neuroscientist Joseph Martinez is investigating how brain chemicals also affect learning and memory.
We find certain chemicals in the brain that we call neurotransmitters. And we think that some neurotransmitters are important for learning in animals and humans. And by knowing what these chemicals are, we are able to mimic how the brain talks to itself. And we can coax the brain into either forgetting an experience, or we can make the brain actually remember better by stimulating some of these chemical systems. In our experiments, we use very simple learning situations. We have a maze which has two arms, and we put the food into one of the arms, and the mouse runs down and obtains the food. The mouse learns very quickly where the food is, and he always runs to that particular arm. Even though we've trained them in this maze task, their memories are not yet fixed. They're not yet permanent. They're not yet into long-term memory, because the brain is still processing this information. And since it takes a period of time to become permanent, we can interfere with this process by injecting the animals with drugs. In this particular experiment, one group of animals we had injected with a drug called scopolamine, which blocked the transmission of information in the brain. And the other group of animals had simply received a saline injection. The animals are trained in a drug-free state. They don't receive drugs while they're learning the maze. When they're finished with their training, we then inject them with the drug, and we find that the drug produces an experimental amnesia. The animals don't remember the maze. They wander around the maze trying to find the food. This is a synapse which contains the neurotransmitter acetylcholine. In the experiment, we used a drug called scopolamine. The scopolamine blocks the receptor site for the neurotransmitter acetylcholine. So in a sense, we've inactivated this synapse. When we do that, it leads to experimental amnesia. We can also do the opposite. We can stimulate this synapse. A drug which does this is called physostigmine. It blocks the enzyme which breaks down the neurotransmitter acetylcholine, so there's more of it around in the synapse to have an effect. When you do this, you actually can produce the opposite result, or enhancement of memory. The reason acetylcholine is interesting in this context is because acetylcholine is the neurotransmitter that's tremendously reduced in Alzheimer's patients. So there's great hope that by understanding this particular chemical system better, that we'll be able to develop drugs that can help people with Alzheimer's disease.